Embryonic development is a remarkable and intricate process, involving the formation and differentiation of various tissues and organs. In this comprehensive script, we will delve into the development of connective tissues, focusing on the mesoderm as the source and the multitude of processes that contribute to their formation and maturation. Throughout this script, we will explore the different cell types involved, signaling pathways regulating their development, migration of progenitor cells, extracellular matrix remodeling, and the role of genetic factors in connective tissue development. We will also discuss the clinical conditions associated with abnormal connective tissue development and their implications in cancer pathogenesis. By the end of this script, you will have a profound understanding of the complex mechanisms underlying connective tissue development. As the mesoderm develops, it gives rise to different types of connective tissues through the differentiation of mesenchymal cells. This involves complex processes such as cell proliferation, migration, and differentiation, which are regulated by various signaling pathways. Connective tissue development is a highly regulated process that involves intricate steps of cell differentiation and lineage determination. The differentiation of mesenchymal cells into specific cell lineages is orchestrated by the sequential activation of transcription factors and gene expression patterns. These factors include SOX9, RUNX2, and ProGamma, which play crucial roles in the differentiation of chondrocytes, osteoblasts, and adipocytes, respectively. Connective tissue cells arise from progenitor populations that possess multipotent or bipotent differentiation potential. These progenitor cells respond to a complex network of signaling molecules, including growth factors, morphogens, and extracellular matrix components. Their fate is determined by the interplay between these signaling pathways, leading to the formation of different cell lineages within connective tissues. Signaling pathways, such as WNT, TGF-beta, and Notch, regulate connective tissue development. These pathways control cell differentiation, proliferation, and extracellular matrix synthesis through the activation of specific genes involved in tissue formation. Overall, the migration of connective tissue progenitor cells during development is a complex process guided by multiple factors including chemotactic gradients, extracellular matrix components, and cell-cell interactions. This intricate interplay allows these cells to reach their final destinations within developing organs and tissues. In summary, ECM remodeling in connective tissues involves the deposition, organization, and regulation of ECM components like collagen, elastin, and proteoglycans. This process influences tissue structure, cell behavior, and mechanical properties, ensuring tissue integrity and function. In summary, the process of skeletal development includes the differentiation of mesenchymal cells into osteoblasts, the deposition of an osteoid matrix, mineralization, and the regulation of bone homeostasis through communication with osteoclasts. This coordinated activity involves various factors and signaling pathways. Cartilage development involves the differentiation of mesenchymal cells into chondrocytes, which produce and maintain the matrix of the cartilage. This differentiation is regulated by signaling events and transcription factors. During cartilage maturation, chondrocytes undergo changes and promote vascular invasion, leading to endochondral ossification. The cartilage matrix is composed of collagens, proteoglycans, and glycoproteins, which are regulated by matrix metalloproteinases and tissue inhibitors of metalloproteinases. The development of adipose tissue involves the proliferation and differentiation of pre-adipocytes into mature adipocytes. This process is regulated by signaling interactions and transcription factors, leading to changes in morphology and the secretion of adipokines. Adipose tissue plays important roles in energy balance, body temperature regulation, and lipid storage. Growth factors and morphogens regulate connective tissue development by influencing cell proliferation, differentiation, and tissue organization. Specific growth factors and morphogens interact with receptors on target cells, triggering signaling pathways that modulate gene expression and cellular responses. Morphogens establish concentration gradients that guide cell fate determination and tissue patterning. The coordinated actions of growth factors, morphogens, and their effectors ensure the proper formation and function of connective tissues during embryo development. Therefore, studying the genetic factors that influence connective tissue development is crucial for understanding the pathogenesis of connective tissue disorders and may pave the way for novel diagnostic and therapeutic approaches in the future. Abnormal connective tissue development can lead to skeletal malformations and connective tissue disorders like Ehlers-Danlos syndrome. Genetic and environmental factors contribute to the development of these conditions. Skeletal malformations affect bone and cartilage, while Ehlers-Danlos syndrome affects various connective tissues, including skin, joints, and blood vessels. Understanding these relationships helps develop targeted treatments. In summary, abnormal development of connective tissues can contribute to cancer pathogenesis through dysregulated signaling pathways, promotion of tumor growth and invasion, and creation of a supportive microenvironment. This knowledge is crucial for developing targeted therapeutic interventions. In conclusion, connective tissue development is a complex process involving mesoderm-derived cells differentiating and migrating to specific locations. Factors such as extracellular matrix remodeling, genetics, and clinical conditions also impact tissue development. Knowledge of these processes is vital for managing connective tissue disorders and cancer.